Hello students, welcome to Lab 5. We're going to be talking about microscopy. Alright, let's get started. Introduction, we've already kind of talked about microscopy in lecture at one point. We have kind of two different general types of microscopy. Light microscopy that has some visibility, some magnification, and then electron microscopy that has a lot more magnification. Light microscopes use light, series of Klein lenses to magnify organisms about 40 to 1,000 times their range. Electron microscopes do a lot more than that. Two different types of electron microscopes: transmission and scanning electron microscopes. Transmission does a lot more magnification than scanning electron microscopes, but we'll do a little bit more talking into that in a minute. All right. So electron microscopes use beam of electrons through a very thin section of the specimen. So you're going to have what's called a microtome. So you have a section of your specimen, and a microtome is a very thin razor, and it just slices the specimen into very thin sections. And usually the specimen is encased in a wax block, and it's just sliced very, very thinly. Sometimes the specimen is whole, and if it's whole, what it needs to do is it'll be encased in a very thin layer of gold particles. But if not, then it'll be thinly sliced in wax. Either way, it will need to be encased in gold particles. Otherwise, what will happen is those beam of electrons will burn the specimen. Because first, what you must do is you have to desiccate the specimen before you put it into the electron microscope. Otherwise, any amount of moisture in there will make the specimen explode. It's not a fun process, and you will destroy the specimen. Basically, you're sending a beam of lasers at it so that you can see what bounces off. So any transmission electron microscope can go into 100,000 times magnification, which is really, really intense. You can see organelles, you can see other things like near atomic size. Useful to see cell surfaces, small organisms, but light microscopes, you can see living things. Electron microscopes, again, it has to be desiccated, and it has to be covered in gold, so it has to be dead. So you can't see anything moving, you can't see anything crawling around, so yeah. All right, the SEM process, once again, you have to slice through anything, and it has to be covered in a layer of gold. So there's a special machine that you can put your specimen in. Usually what you do is you tape your specimen to a knob piece, and then you put it into the machine, and it just layers everything in a fine coating of gold. And then if you see at the top, there's this just electron beam that scatters electrons everywhere. And then whatever bounces off, there's these detectors all around inside the machine that will detect how or where those electrons bounce off. And it'll see what angles those electrons are coming from, and then it will see how the specimen is shaped, and it's really cool. I've only got to do it once, and I did it with cactus pollen, cactus spines, and other things. So, all right. And then the light microscope, the ones we would have used in lab, those are pretty much used in most universities, most colleges, because they're mostly used for learning, and they're pretty widespread. So we can see cells, we can see some organelles like nuclei, some stained cells, cell organelles like mitochondria, chloroplasts, other things like that. So the light comes from the base, the light source, it gets polarized by the diaphragm, comes through your stage where your specimen is, and off to the side you can see some plant cells, and on the top of that, that's some animal fat cells. And then it goes through some different lenses, goes into the ocular lens, and then you can see it with your eyepiece. Now, as it's going through those lenses, the light has to switch places, right? It has to fold and magnify, right? So what does that do to the image? Well, it's what's called inversion. So the reversal of the image of the specimen by those lenses. So usually what you'll see is it's going to flip the image upside down. It's going to actually turn it upside down. So if you saw an F in there, you'll see it turned upside down, right? If you put an F basically facing up on your slide, if you saw it in the image, it would be upside down in your ocular lens. So when you move the image on your stage, it would move the opposite of how you would be moving it on the stage from what you were looking at in your ocular lens. So keep that in mind whenever you go into labs later on in your biology or later sciences. Okay. All right. So when you look at the microscope itself, you have a few different parts. The main parts I want you to focus on are that stage clip, right? So you're gonna have your slide, and the main way that you're gonna put your slide on is you're gonna need to fold out that stage clip, put the slide in, and then put the stage clip back on. Now, that may seem second nature to a lot of you, but some people have never used a microscope before, right? Some people actually try to pull the stage clip up. Never do that, right? Don't pull the stage clip up and try to shove the slide underneath. That breaks things. Which is another thing that you should never do. Never start with the most, or with the highest objective lens, right? You always want to start with the lowest objective lens. So we have 3, sometimes 4 different objective lenses, right? You have the 4 times or what's called the scanning objective lens. It's usually red. We have the 10 times, what's called the low power objective lens. We have the high power, or 40 times objective lens, and we have sometimes the 100 times, but we don't use that one, right? That's called oil immersion. We don't really use that one in this lab. If you go into later labs and they tell you to use that one, fine by me, but we don't really use that one in this lab. So we'd focus on those three, right? You always want to start with a scanning objective lens in any lab, right? Anytime you start with a new slide, start with a scanning objective lens. Why? Because you don't want to break things. So, start with the scanning, pull out the stage clip, put your specimen in, put back the stage clip, it should lock in place, right? And then what you want to do, you want to start with a coarse adjustment knob. Coarse adjustment knob is on the inside. Look inside, see if you can focus, right? If you're not close enough, you're going to want to pick up the whole microscope. You don't want to pull it across the table like it's going to rattle, right? If you rattle things in there, then it might break something inside. You want to pick up the microscope and move it closer to you. Pick it up by the arm and the base. Don't pick it up by anything else. Okay. Adjust the coarse focus knob. And then try to find out where your specimen is, try to focus on it, and then you can move your objective knob. You don't need to refocus anything. Don't need to move the stage, don't need to move anything else, because these are what's called parafocal lenses. Whenever you switch a lens, it should remain in focus, right? So you switch to your low power, which is the yellow one, right? And it should remain somewhat in focus. The only thing that you should need to do is adjust some fine focus on it. And then if you need to switch to the high power, you can switch to the high power. Again, don't move the stage, don't move anything else if you need to. And then observe whatever you need to do. Keep in mind that as you switch objective lenses, your light source is going to go down, because as you zoom in, there's less light coming in to your focus. So you might need to adjust that light source a bit. Let's see what else. The next thing, do not use the coarse focus whenever you have that high power objective lens on. Why? Because if you adjust that coarse adjustment knob, if you have that high power objective on, it's going to pull up and it's going to break right through that. And you don't want that to happen. It's going to break the objective, which is really expensive. It's going to break the stage, again, really expensive, or it's going to break the specimen, which sometimes can't be replaced. All right. Objective lenses, magnifications, we kind of went over this. So the objective lenses, scanning is 4x, but with your ocular lens, that's 10x. So 4 times 10 is 40. Low power is 10, times 10 is 100, and so on and so forth, right? 40 times 10 is 400. because your ocular lens is 10x, you have to multiply your objective lenses by your ocular lenses. All right. Review for practical 1. Just some more questions, things that I went over on here. Function of the diaphragm, just an adjustment to the light on the bottom. Anything else on here should be in your lab manual. All right, I believe that would be it.